Good afternoon, everyone. I am Dr. Kathleen Tate, Editor-in-Chief of the Journal Online Learning, Research and Practice, and Department Chair and Professor at American Public University System, and will be the chair of this session. It is my honor to introduce this young man, Mr. Balin Ratliff, Senior at the University of Washington in Seattle, an invited student speaker. He is studying marine biology and is a Mary Gates Endowment Scholar and a Noah Hollings Preparatory Program Scholar. In summer 2023, Balin was accepted into the National Science Foundation Research Experiences for undergraduate programs at Friday Harbor Laboratories. There, his research focused on identifying resilience in eelgrass against seagrass wasting disease around the San Juan Islands, Washington, aided by the AI program ELISA, eelgrass lesion image segmentation application. He just returned recently from another research trip to Costa Rica. Balin's session title is Leveraging AI to Conserve Seagrass in a Changing Ocean. It is my pleasure to turn it over to Balin Ratliff.
Yeah, thank you for having me. So, like Dr. Tate said, I will be talking about eelgrass. And I understand that this is probably the first time many of you have been introduced to them. So I would love to set the stage and just talk about them for a little bit. So yeah, welcome to Leveraging AI to Conserve Seagrass in a Changing Ocean. First off, I'll give a background of our study area. So this is Washington State, all the way in the Pacific Northwest. I know many of you are coming from the East Coast, so just a little bit away. And this green box is part of the Salish Sea. The Salish Sea encapsulates all of the Puget Sound, so all of Seattle area, further up into British Columbia. And this is really a really cool area where a bunch of life lives. And I like staying here because I get to study all of it. Specifically where I was studying is Friday Harbor Laboratories in the San Juan Islands, which is a really cool place where you just get to do a bunch of work. Students and faculty there are amazing. It's a UW institution that's been there for over 100 years. And I've just been really focused on the eelgrass stuff there recently. And I'll just talk about what I did for the past year, last summer. First off, I want everybody to just think of a rainforest and how beautiful and amazing and diverse those ecosystems are. All the beautiful birds, jaguars, all the cool things. And just think about what makes that so special. Like, what makes that environment? And of course, it's the trees, right? So now just imagine putting that underwater and boom, you've got eelgrass. Eelgrass is super important. It creates the environment that you see right here, and really makes life possible for an endless number of creatures. We know it as a key ecosystem engineer, and what that means is it makes the environment. So it cleans water, it stabilizes sediment, it creates habitat for crabs, fish. If you know how important salmon is to us, these guys are incredibly important for salmon and their prey. So all around super important species. And just a little fun fact, these guys are a grass. So they're related to terrestrial species, but now they live in the water. So that's just a really cool thing about their evolutionary pathway. And we know that they are very essential to the Salish Sea. A bunch of Washington Department of Natural Resources reports and background studies really have classified this as a super important living thing that makes life in the Salish Sea possible. Sadly, of course, I have to get to some bad news. It's declining in the San Juan Islands. Out of all of the Salish Sea, from the studies we've done, this is the area of most concern, and you can see that here in these figures. On the left side, we have long-term trends for 20 years of study, and we can see that for a large majority of the areas with notable trends, we see declines. And that's all that red bar is, with only some small declines at 3 sites. And then our recent trends for just the past 5 years, only declines for the ones we can determine trends of. And that's really devastating news if you care about salmon, if you care about orcas, if you care about everything on our coastline, including just protecting it. Like I said, it stabilizes sediment. So it's really important for humans as well. But luckily enough, we know that it's important, and the Washington State Senate has already set aside 10,000 acres of area for us to conserve kelp forests and eelgrass by 2040. So that's some good news. We have something to work towards. We have area set aside, but we really need to know how to use that space effectively, right? And there's even some more good news. So we've already made some leeway on getting protections done for kelp and eelgrass. This is one area near Everett, but that's only 2,000 acres. When you look at it from the top down, not that much area compared to the entire area of the Puget Sound. So looking forward, we really need to understand why these declines are happening so that we can make sure we're meeting the needs of eelgrass. So what is one of the main issues? That would be seagrass wasting disease. It's caused by a little protist called Labyrinthula zosterae, which our lab affectionately refers to as Labby. And it's a parasitic protist, microscopic, and it's opportunistic. So that means that when eelgrass are vulnerable or weakened, that's when the parasite really likes getting in and doing its damage. It's also ubiquitous, so it's almost always in the water column. No matter where you go around the San Juan Islands or Puget Sound, there's probably some Labby in there. So the real concern is what's happening in the environment that's making these eelgrass more vulnerable to these parasites. And moving forward, I just have two terms to define so that we're all on the same page. Prevalence is the percentage of a population that contains the disease. So that's how we get a sense of disease distribution over an area. And then severity is the total percentage of lesion area on a leaf. That's how we know how badly an individual plant is infected. And those lesions look like this, just these little black dark marks on the blade of a plant. And all of that is necrotic tissue. The parasite just loves to get in, munch on some cells, and just leaves the dead cells there. And for some cool figures that also share some bad news, we're all not strangers to climate change. On the left figure, you see that wasting disease lesion area, which is severity. And then on the x-axis, we have cumulative positive temperature anomaly, so increased temperature. You can see that increased lesion area or increased severity is correlated with the cumulative positive temperature anomaly. So you can see that with increased warming, we expect to see more severe infections. And on the right figure, we see that blade growth on the y-axis and disease severity on the x-axis, when it's more severe, there's less blade growth. So we have kind of a feedback loop here of vulnerable eelgrass get infected. Under climate warming, the disease gets worse. And when the disease gets worse, the plants are less able to survive and thrive, which down the line impacts everything that relies on it. So our study is really focused on using disease as a proxy to try and find if there are resilient meadows. And my hypothesis is a opportunistic one. I'm going to say yes, certain sites around the San Juan Islands are more resilient. And then what do we mean by resilient? So resilience is determined by multiple factors. What I'm really most interested in are the abiotic factors, the environmental factors. So there's something like depth. Less deep eelgrass may interact with humans more, with boat propellers, swimming, whatever. These are very delicate plants, so the more you interact with it, the more they'll be disturbed. And then there's water flow. Like I said, Labby is just in the water. So maybe the amount of time water is in a certain space impacts how easily they can be infected by the parasite. And then there's turbidity. We know that eelgrass, they need to photosynthesize, but increased turbidity means the water is really cloudy. That means they get less light and are less able to produce sugar, produce energy, and are less able to defend themselves against the disease. So these are all abiotic stressors that may have a role in predicting or detailing the context of resilience and infection. And then the couple biotic factors that the Harvell lab is interested in are genetic diversity and the microbiome. We won't get into that today, but they are both really interesting things that we're looking into in the future. So let's get back to the question. Are certain eelgrass meadows resilient against these stresses? So what did we do? We went out with a really cool team of community scientists and local experts from Friends of the San Juans, really cool people. Work wouldn't have been possible without them. And this was basically a 3-part process. So at the start, we would go out to a predetermined site at low tide to make sure we could snorkel and get in there without, diving way too deep. And then step 2 was just to collect plants. We would go out there, try to random sample some plants, pick up one every foot or so, and just collect 20 per site, put them in a bag with seawater, and take them all the way back to the labs. And part 3 is just sit down and measure. So we would get all the plants, we would measure their length, the leaf length, leaf width, how many leaves were on a plant. So we get all these biometric data that allow us to contextualize other disease metrics and understand the health of the plants. And this is the last step of that part 3. In the lab, we like to scan it so we can get any blades that we think have disease on them, those little black lesions I showed you earlier, that we can have a record of them, and boom, we have it here, and we label it 1, 2, 3, and 4, each for a different plant. And if you can imagine, in the past, how did we quantify disease area? Researchers would just have to look at a blade and determine, okay, this is a lesion, this isn't, and then calculate severity that way. But even looking at this yourself, you can tell that can be pretty difficult. All of this brown and black splotchiness, that's not all disease. So how do we really make that more efficient if we need to know accurately how much disease there is and, just have it for a record? Well, this is an AI conference after all. So disease analysis was performed by ELISA, our really cool eelgrass lesion image segmentation application, developed a couple years ago on thousands of over 1,000 training and testing scans, like the one I showed you prior. And what this does is it allows for a standardized disease evaluation. Instead of researchers having to be trained on specific techniques and different labs across the world having different determination, we can just put it through this really sophisticated system and have a consistent box where we can say, this is what the disease looks like. And it's actually so much faster. It's 5,000 times faster than human researchers, and it has comparable accuracy. So it's really a no-brainer to start using this program to quantify disease. And it's really cool. All you have to do is put in that scan, and this is the scan I showed you earlier with these 4 different leaves, and it gives you this output. The green is all the healthy tissue it's determined, and the red is all the necrotic lesion tissue it's determined. And from this reading, it automatically gives us a prevalence, and it gives us our severity. So no measuring and making sure to get it to the closest hundredth of a decimal. The program does it for us, and we can understand what the disease looks like that. So what did we find? Well, we surveyed 11 sites across the archipelago, which was no small feat. Once again, thank you to the volunteers, which was over 250 plants, which is a pretty good size. And I want to give some attention to these different sites. Like I said before, abiotic factors likely have a role in resilience. So I wanted to bring attention to the fact that these sites were very different. At Mud Bay, you can see me snorkeling there. It might be 2 feet deep at most, and I can barely see in front of me because of how turbid the water is. But Beach Haven is relatively clear. It was a little bit deeper, and you can see that the plants are pretty long. So really different abiotic features. So the first result I want to go over is prevalence, just where is the disease and how much is there? Well, we can see here that it's not a uniform distribution, which is pretty interesting because we know it's ubiquitous in the water column. The disease is everywhere, but we can see that the infection is not necessarily everywhere. Specifically at False Bay and Beach Haven, we found no disease on our 20 plants, according to ELISA. And that's really interesting. So maybe there's some resilience happening there. But like I said before, severity is really the thing that impacts it most. It's a severity-dependent disease. So we really need to understand where is the disease most severe as well. And as you can see, the prevalence and severity of the disease are not comparable. We can see that severity is pretty low across the board at many places, but Fisherman's Bay is ridiculously high comparatively. So we also understand that disease severity differs at different sites, which also lends evidence to suggest maybe there are resilient areas in the San Juan Islands. And if we can find out why that exists, we can actually get to the bottom of what we can do to help, and how to conserve sites that have a chance really against climate change and future human disturbance. And to go over that, conservation is time sensitive. We know research takes a long time. This graphic I developed doesn't even fit all the things we need. It doesn't fit the grant proposals, or any of the outcomes, really, the conservation outcomes. This is just the work. And when we're thinking about the work, there's a lot that comes after it. But while we're doing all of this, climate change, human disturbance, pollution, this is only anticipated to increase. So we need some way to do all this faster so we can get ahead of the curve and really make good decisions with these acres we've been given to do conservation. And ELISA allows us to do that. So we have faster data processing, which becomes faster analysis, and then we're able to act faster in the face of all this climate change and all this other environmental things that are really concerning for eelgrass. Basically, in conclusion, the speed of ELISA, it helps us save eelgrass. So using ELISA, disease data can be processed quickly and accurately, 5,000 times faster than a researcher. It's accurate and it's standardized. So anyone in the world can use that program and make it go the same way. And we also can estimate site-specific resilience by using disease as a proxy. Resilience can be really hard to grasp if you don't have a metric to measure against, but by using disease, we're able to do that and quickly with ELISA. And since meadows with higher severity are less resilient, we can start looking into why that is. What conditions exist in these spaces that make eelgrass vulnerable, and then use that information to decide, okay, these areas are really vulnerable. They might need more help, or we need to focus on these other areas that have more resilience against climate change so that we can start planning for the future. And really, again, this is coming back to we already have 10,000 acres guaranteed for kelp forest and eelgrass meadows. We just need to know how to spend those acres the best. So ELISA's helping us do that, and I'm excited to help save eelgrass. Just to review a couple of other things, I wanted to bring attention to the Department of Natural Resources. They're leveraging AI as well for a related project concerning eelgrass. They're using towed underwater videography, which is they walk a transect with a boat, and then they record the bottom. And what they're trying to do while collaborating with Microsoft is use an AI to identify specific species and area. So here you can see that just under a performance test, they did 15 sites and over 9 hours of footage. And their AI program has already been almost 95% accurate just looking at video footage. So I think that's really cool, and that will speed up our ability to understand how eelgrass is doing on a larger scale, not talking about disease, just area distribution. So I thought that was really cool, and I wanted to bring that up. It's not just us using AI. DNR is also using AI. And then also in other areas of the world, and other things I've noted, there's automation AI everywhere. So there's automated drone flight patterns. You can program a drone to fly a specific path and record directly down, and you get high resolution imagery. It's really quick and it's very reproducible because you just fly the same path every time. So that's a really interesting new area of expertise that's coming about. I know Dr. Corey Garza at UW is doing that in California. And then from my time in Costa Rica, this was a picture from then. They're doing it in a little estuary area, and they're able to see sharks from the top. It's really cool doing it that way. And then just a few other things, satellites and AI. I know there's groups using it to count whales from space so that you get an abundance estimation and area use determination. And then there's detecting plastics from space so you can see where it's going. And then huge area for AI up and coming is fisheries management. Really complex databases, a lot of moving parts, and again, an area where time constraints are a thing. So AI's speed and efficiency is really what's super valuable in these spaces where we need to make decisions quickly, but also those decisions need to be well informed. And that's it. Quick review of references, and then yeah, thanks.
Ba- Balin, what an outstanding presentation. We have lots of questions, so we'll see how far we can get with them. Let me pose the first one, and this is keeping in mind you were studying marine biology, but maybe you can answer this question. Disease and water infections threaten meadows. So can you extend beyond seagrass studies and research with AI to explore other parts of the environmental animals that need meadows and water? What about using your AI experience to explore reindeer and caribou translocations across the circumpolar north? And maybe be able to study how people and reindeer have shaped each other in times of rapid environmental and political change.
Oh, absolutely. I mean, that's completely outside of my expertise. And the AI portion of this was not my doing. I simply used the application. It was a really useful tool. But I mean, AI has its uses in many places. I think that's a really interesting question about reindeer and human interaction. Uh, Not something I could really speak on, but yeah.
Here's another question. It says, this is so amazing. If we can really have good applications to make life and the environment more healthy, how do we ensure that the knowledge is accessible to poorer parts of the globe?
Mm. Okay. Yeah. How do we make it more accessible? I mean, hopefully talks like this make it more accessible as well. I'm a huge outreach proponent. I love talking to people and meeting them where they are. And I think as far as AI go, as long as we're getting data faster, so then we need to know how to share the data faster and how to talk to other people and communicate it. So it's not only the AI that needs to advance, we as researchers need to advance and how we get that data and information across as well. I can't really speak on how, but I know that I really want to do that. Yeah.
That's excellent. Here's another question. Good to see that AI is allowing for a speedier process for conservation. Did you or your team encounter any problems with ELIZA?
Okay, yeah. So this was an actually really interesting thing that happened. I'm glad you asked. I kind of took out a slide of here. I wanted to talk about it, so I'm going to now. Uh, so we ran the process 3 times. The first time we got the information back, like you saw with the 2 sites that had no prevalence. That was kind of unprecedented in the research that had been done prior to me. So I looked at that with instant skepticism. So I told my PI, Olivia Graham, I said, hey, can we rerun this scan? So we did. And then we got vastly different information, but prevalence was all consistent across the board. So I said, okay, this makes more sense. Then 2 months later or so, we went back and looked at the scans we got and looked at the severity and prevalence reading from ELIZA and said, that doesn't line up at all. What was happening? And while we still don't really know, we ran the scan again, and it was almost directly proportional to the first scan we did initially. And then we went back and looked at the scan and said, yes, this does make sense. It's weird that prevalence isn't at these 2 sites, but it's not, based on the plants we collected and based on ELIZA's look and based on our own expertise. So ELIZA wasn't necessarily the problem there. There was a little bit of a weird change with that second scan. We don't know why it processed them very differently. So a word of caution to make sure you run your program multiple times to make sure it makes sense. But that's also like an informative thing for me to not let my judgment cloud and think, oh, the program got it wrong. This can't be right. So that's something I had to recognize in myself, just to be like, I can look at it and make sure it's right or wrong. My gut instinct was wrong in that instance. So I think that's a really interesting interaction between our human side of research and then our more technological machine learning side. And we need to try to find a balance of criticizing that, but also finding its utility and sometimes trusting the results.
No, Balin, those are really great points. That's comes up with AI as a tool. We have to be discerning. We have to teach people to have discernment and really look at things and, like you said, evaluate them with thought and check what's going on. Here's another question. I don't know, this might apply in marine biology since there are sea creatures, but the question is there a connection between your work and wild animal deaths or sickness?
I would say no. As far as I understand, we don't know a lot specifically about Labby's life cycle at all. But as far as I understand, it doesn't infect other species or animals for that matter. So in that sense, we're not studying an organism that would impact a higher trophic level directly. Um, and ELISA, as far as I understand, since it's only measuring disease in a very specific way on eelgrass, it wouldn't really help us track anything traveling up a food chain terrestrially, if that helps.
That's very helpful. I don't see any other questions remaining. So at this point, we will close the session and give you a big thank you for sharing about this. There are applications across, well, within science, beyond, and just, how to use AI as a smart tool. I like what you said about faster data processing, faster analysis, and faster action. I mean, those are very important things. So thank you so much, Balin Ratliff, and everyone else. We have time for a short break before the next session.